We now consider the class of lipids known as the steroids. And steroids, by definition, are molecules containing a tetracyclic 6-6-6-5 ring system in the orientation shown on the right side of your screen. And there are 17 carbons in this core ring system. And the carbons are numbered as shown, starting with carbon 1 on the left side and ending with carbon 17 on the right side. So you don't actually go completely consecutively around the ring system, but what you're actually doing is numbering the left-hand ring first, then the next ring over, then the next ring over, and then finally the right-hand ring. So these rings are commonly referred to as the A, B, C, and D rings. So steroids are actually biosynthesized from terpenes, which we've already talked about. So in order to make all of the branching work, real steroids have methyl groups at C10 and C13. And then all natural steroids have some sort of oxygenation, so either an alcohol or a ketone at carbon 3, and then something else, whether it's just an oxygen atom or a side chain at carbon 17 coming off of the D ring. Okay, so I want to start by looking at some of the steroids that are present in the body, and the parent of this whole class is cholesterol. So cholesterol is a structural lipid, so we could have talked about it in a previous lesson, in that it adds rigidity to cell membranes. Okay, if you just had the phospholipids, cell membranes would be a little too squishy, and having some cholesterol molecules in there being a rigid tetracyclic structure holds the membrane kind of in place where it belongs. Now, of course, cholesterol is pretty famous these days because we all know that if you have too much cholesterol traveling around your bloodstream, it will start to deposit on artery walls, leading to hardening of the arteries and eventually potentially heart attacks or strokes. Now, cholesterol is also important, not just in and of itself, but it is actually the precursor, the biosynthetic precursor to other steroids in the body that we'll look at on the next slide. So the body actually uses cholesterol as a starting material to make whatever other steroids it happens to need at a moment. So what are some of these steroids? Well, the sex hormones are a very important class of steroids. You have the male sex hormones, testosterone being the most important, but also androsterone. And then the female sex hormones, there are actually two compounds that together are known as the estrogens. One of them is estradiol, in which you have a hydroxyl group at the 17 position. And the other is estrone, where the 17 position is oxidized to give a ketone. Then finally, in progesterone, you have two extra carbons coming off of the 17 position. And what amazes me when I look at these structures is, look at how similar the structure of progesterone and testosterone are. One is a female sex hormone, the other is a male sex hormone. The difference between them are two carbons at the 17 position. So a couple of other steroids that are important in the body are cortisone and cortisol, which are, of course, synthesized in the adrenal glands, and they are responsible for the body's response to stress, what is typically thought of as the fight or flight response, which results in an elevation of blood pressures. Sometimes cortisone itself is given therapeutically as an injection, but more commonly, the anti-inflammatory steroid that is administered as a drug is called prednisone. So it looks a whole lot like cortisone. The only difference between them is an extra double bond in the A ring in prednisone. Prednisone can be taken orally, and its function is to actually prevent the release of arachidonic acid. And as we're going to look at in the next lesson, arachidonic acid gets cyclized to give prostaglandins, which are responsible for the inflammatory response. So once we start talking about the steroid rings, we have to take a moment and look at conformations and also the new topic of ring junction stereochemistry. So the 10-carbon molecule with two 6-membered rings fused to each other is called decalan. And I'm just looking at that as a simpler example to show what I'm talking about. So between two 6-membered rings, you can either have a cis ring junction or a trans ring junction. So what that of course means is it's the question of whether the hydrogen atoms that are present at the ring junctions are cis to each other or trans to each other. The cis-trans relationship is easiest to see in the planar drawing, but you have to remember that these molecules really exist in the chair conformation. So in the trans ring junction, both of the hydrogens can occupy axial sites because one is up and one is down. You want the smaller hydrogen atoms to occupy the axial sites because the other substituent at those carbons is the other ring that's being fused to the ring that you're looking at. And it, of course, is going to be bigger than hydrogen atoms and will want to be equatorial. For the cis ring junction, in order for both of the hydrogens to be up, one of them can be axial, but the other one has to be equatorial. Therefore, for a cis ring junction, the entire structure is what we call concave. The trans ring junction is more aesthetically appealing because it leads to a nice flat structure. Okay, so let's take these ideas and apply them to steroids. So in virtually all steroids, the BC and CD ring junctions are trans, but the AB ring junction is sometimes cis. Okay, so first I want to look at a trans AB ring junction. So this is the three-dimensional chair cyclohexane view of a steroid with a trans AB ring junction. So in this case, we know that there's a methyl group on carbon 10. So in this case, it's the hydrogen on carbon 5 and the methyl group on carbon 10 that are trans to each other. And sometimes steroid chemists call this the 5-alpha form because the hydrogen on carbon 5 occupies the axial site, which if you think back to carbohydrates, at the anomeric carbon, the alpha site is an axial site. Okay, so the hydrogen here occupies an alpha site, whereas in a cis AB ring junction, the hydrogen on C5 and the methyl on C10 are cis to each other, and this structure is called the 5-beta form because the hydrogen on C5 occupies the equatorial beta site. Okay, so that's a little bit of the structure and function of steroids. I want to take just a couple of minutes to go over the very broad overview of how nature makes steroids. And I'm going to focus specifically on the biosynthesis of cholesterol because, as we know, other steroids come from cholesterol. So cholesterol is actually synthesized from a molecule called squalene, which is a triterpene. It's a 30-carbon terpene, and it's all carbons and hydrogens. Now, as you are aware, terpenes are made from plants. Humans do not actually synthesize terpenes, except for squalene, which is used to make cholesterol. And humans, instead of using dimethyl allyl pyrophosphate and isopentenyl pyrophosphate to make squalene, humans actually synthesize squalene from something called HMG-CoA, which stands for hydroxymethylglutarate coenzyme A. Alpha-ketoglutarate is one of the intermediates in the citric acid or Krebs cycle. So you might have heard of the statin drugs that are commonly used to treat high cholesterol these days. And what they actually are inhibitors of an enzyme called HMG-CoA reductase, which reduces the ketone in HMG-CoA, and that is the first dedicated step towards the buildup of squalene and eventually into cholesterol. So the big picture of the conversion of squalene to cholesterol, what I want you to get out of this and the next couple of slides, is that each of the 4 rings is closed in sequence, the A, the B, the C, and then finally the D. And then there are some rearrangements and cleavages to end up at the required 27 carbons with every functional group in the proper place. Okay, so starting out with squalene after several steps, we end up with a carbocation. So the general way that rings are closed are what are called Cation Pi Cyclizations, where you have a carbocation, which in this case is tertiary, and it is attacked by the pi electrons of a double bond such that you close the 6-membered A ring. Okay, so that's the first ring closure. And then moving forward, you have double bonds spaced in exactly the right place. These double bonds, of course, all originating from the squalene, so that you can do the cation pi cyclization again to close the B ring. So that takes us from the top structure to the next structure down. We do it once more to close the C ring. That takes us one more structure down. And then finally, we close the D ring. And then the rest of the squalene serves as the side chain coming off of the 17 position of the ring system. And then I'm not going to actually say anything else about the rest of the biosynthesis, because from this point onwards, it's all rearrangements, proton transfers, and then other functional group manipulations to get you to the final product. Okay, so that's our brief overview of steroids. And as I mentioned very, very briefly in the context of prednisone, another class of lipids that we have to consider are the prostaglandins. And that's going to be the subject of our next lesson.